Hello, and welcome to our series, What to Look For, where we talk about specific considerations for investigating foodborne illness outbreaks. In this video, we will discuss specific considerations for investigating Campylobacter outbreaks. First, let's go over some of the basics of Campylobacter. Campylobacter is a bacterium that can infect human cells and cause disease. Campylobacter is microaerophilic, which means that it can only grow under highly specific oxygen and carbon dioxide requirements, for example, the conditions found in the gastrointestinal tract of animals that hosts this pathogen and represent its reservoirs. Because of its microaerophilic nature, Campylobacter can survive, but is unlikely to grow in many environmental reservoirs that do not have these specific oxygens and carbon dioxide conditions. For example, Campylobacter can survive, but is unlikely to grow in environments such as water, soil, or on plants. On the other hand, specific animal reservoirs, such as chickens, cows, ducks, and many others, are able to support both the survival and growth of Campylobacter. Because of this, most foodborne illness cases and outbreaks associated with Campylobacter are highly specific to certain animal-associated food products like raw poultry and raw milk. Campylobacter causes an estimated 1.5 million cases of foodborne illness in the U.S. each year. Common symptoms of campylobacteriosis include abdominal cramps, fever, vomiting, and diarrhea. The typical incubation period of Campylobacter is 2 to 5 days, and the disease is generally self-limiting, with symptoms typically lasting between 2 and 10 days. However, in a handful of cases, persons with Campylobacter infections may develop a disease sequelae known as Guillain-Barré syndrome, which is a long-lasting nerve disorder that can cause muscle weakness and paralysis. Common local establishments where Campylobacter outbreaks can occur include restaurants and other eateries, long-term care facilities, schools, and penitentiary facilities. When investigating Campylobacter outbreaks linked to local establishments, there are a variety of areas where environmental health investigators may need to focus their investigation efforts. These include evaluating whether improper cooking practices have contributed to the outbreak, evaluating if cross-contamination of Campylobacter during food handling or storage could have contributed to the outbreak, as well as evaluating whether contaminated food products or ingredients may have been the source of the outbreak. First, let's discuss how to evaluate whether improper food preparation practices, such as cooking, could have contributed to the outbreak. Retail and food service establishments that prepare food need to make sure raw foods of animal sources like poultry, meat, or egg products are cooked to proper temperature and time to ensure that any Campylobacter that is potentially present on these raw ingredients is inactivated or killed. Remember that, although the poultry industry is doing everything to eliminate Campylobacter from raw foods like poultry and eggs, there is always a chance that Campylobacter could be present on these foods before cooking. For example, in a 2020 Campylobacter outbreak associated with chicken liver at a restaurant in Washington state, environmental health investigators identified that the chicken liver mousse prepared at the restaurant was not being cooked at an appropriate temperature that is effective for inactivating Campylobacter. To evaluate whether improper cooking practices could have contributed to a Campylobacter outbreak, environmental health investigators should identify whether any cooking practices used in the establishment were not appropriate to ensure inactivation of Campylobacter. These processes should include reviewing relevant records in the establishments, such as 1. Standard operating procedures, such as SOPs or recipes, and 2. Documents where monitoring and verification activities are recorded. By reviewing the SOPs, the investigator is making sure cook procedures are appropriate, and by reviewing the monitoring and verification records, the investigator is making sure these cook procedures are consistently being followed. In addition, investigators can also review other documents that are relevant. For example, logs that show how often thermometers used to monitor the cook step are calibrated, and what calibration records say about the accuracy of the thermometer. To supplement this review of records, investigators should also conduct interviews with both management and food handlers at the establishment and ask questions about 1. How SOPs for cook steps are enforced, 2. Any potential inconsistencies in SOPs and monitoring or verification records, as well as other questions about 3. Food thermometer calibration and maintenance. Whenever possible, investigators should observe or ask food handlers to demonstrate the cooking procedures. Next, let's discuss how to evaluate whether cross-contamination of Campylobacter during food handling or storage could have contributed to the outbreak. Campylobacter can spread from contaminated food products to non-contaminated food products if food handlers are not washing their hands or changing gloves between handling foods like raw poultry or meat and handling ready-to-eat foods such as fresh fruits and vegetables. Also, not properly cleaning and sanitizing equipment and utensils between handling different types of foods can also potentially facilitate cross-contamination of Campylobacter. Additionally, poor separation and storing foods like raw poultry or eggs above ready-to-eat foods in refrigeration units can, in some instances, facilitate cross-contamination of Campylobacter, as the juices from these raw ingredients at higher risk for being contaminated with Campylobacter can drip down onto these ready-to-eat foods. To identify whether food handling or storage practices could have facilitated cross-contamination of Campylobacter, environmental health investigators should evaluate the practices used to prepare and store food products in the establishment during site visits. This can be done through observing how food is routinely handled in the establishment. During these observations, one key thing to take note of is the accessibility of hand-washing sinks and gloves. For example, are hand-washing sinks properly stocked with soap and paper towels? Are gloves available and easily accessible to food handlers? How frequently are employees washing their hands or changing their gloves? Are food handlers changing gloves between handling raw ingredients and handling ready-to-eat ingredients? Additionally, pay attention to how equipment and utensils are segregated. Are food handlers cleaning and sanitizing equipment and utensils between handling raw ingredients and ready-to-eat products? Or is there a color coding system in place? For example, are different color cutting boards used to handle raw ingredients than other food ingredients or products? Also, note how food is stored in refrigeration units. Are any raw ingredients stored above ready-to-eat food products such as fruits and vegetables? Are all raw ingredients stored in leak-proof containers? Such observations are crucial, as failure to properly segregate raw ingredients from ready-to-eat foods during handling or storage can increase risk of Campylobacter outbreaks. For example, in a 1997 Campylobacter outbreak associated with sweet potatoes served at a senior center in Connecticut, environmental health investigators observed food handlers handling raw meat and poultry products in an area that was usually reserved for handling cooked foods and raw vegetables, which could have facilitated the risk of contamination of the sweet potatoes with Campylobacter. Additionally, investigators can also conduct interviews with both management and food handlers about these routine food handling and storage practices. Important questions to ask during these interviews include 1. How is hand washing enforced in the establishment? As well as 2. Are food handlers encouraged to wash their hands or change gloves between food handling activities? 3. Are there instances in which food handlers are advised against using hand washing sinks or changing gloves due to scarcity of supplies? Also, it is important to ask food handlers on how they have been trained to properly segregate raw food ingredients from ready-to-eat products and how these segregation practices are enforced by management. Finally, let's discuss how environmental health investigators can go about evaluating whether contaminated food products or ingredients may have been the source of the outbreak. In certain instances, food ingredients may have been contaminated with Campylobacter before they are purchased or obtained by an establishment. In particular, when investigating an outbreak associated with a single local establishment, these contaminated ingredients are often sourced from local farms or small-scale local producers. Examples of some of these food products or ingredients that might be at high risk for contamination with Campylobacter before they arrive at the establishment include raw milk and eggs, particularly if they are sourced from local farms. To evaluate whether contaminated food products or ingredients may have been the source of the outbreak, environmental health investigators may want to collect select food product or ingredient samples from the establishment and test them for the presence of Campylobacter. Some key things to consider when collecting food product samples include making sure that sterile or sanitized tools or utensils are used to collect samples. For example, if you are using utensils, such as knives or tongs, from the establishment, be sure to clean and sanitize them before collecting food product samples. Additionally, it is important to ensure that all collected samples are transported to the lab appropriately. More specifically, as Campylobacter is sensitive to atmospheric oxygen, proper sampling and packaging of samples to restrict access to atmospheric oxygen should be considered. For example, minimizing headspace in a container when sampling liquid samples or sampling a larger piece of solid sample that is harder to be oxygenated in the center rather than cutting the larger piece into small pieces. Additionally, for certain highly perishable products such as raw milk, samples may not be available to be collected from the establishment. In such cases, collecting samples from earlier stages of the supply chain, such as from retail stores or farms, may be necessary. For example, in a 2015 Campylobacter outbreak associated with raw milk produced at a local dairy farm in California, environmental health investigators at the California Department of Public Health collected and tested raw milk samples produced by local dairy A as part of an outbreak investigation, including 9 bottles from several retail stores in the area and 11 bottles directly from local dairy A's farm store. None of the 9 bottles collected from the retail stores in the area tested positive for Campylobacter, while 3 of the 11 bottles collected directly from local dairy's A's farm store tested positive and yielded the outbreak strain of Campylobacter. The key observation here is that when conducting product sampling for an outbreak investigation, samples may need to be collected at multiple locations where food product is sold. In summary, in this presentation we have covered some of the specific characteristics of Campylobacter, as well as 3 key areas that are relevant for environmental health investigators to consider when investigating Campylobacter outbreaks linked to local establishments. In addition to Campylobacter, check out our other videos in this series that cover key considerations for investigating foodborne illness outbreaks linked to other foodborne pathogens.